talk is disabling melanopsin and its non-image forming output in the mammalian retina.
All right, so my name is Sam Luebbert, and for the past 2 years, I've been working in Professor Provincio's lab upstairs. And today I'm showing you a project that I've been working on the past 6 months. And it's an attempt to disable melanopsin in the mammalian retina. All right, so here's some background. So what you have here is a cross-section of the eye. So light comes from this direction, strikes the cornea, passes through the lens and the pupil, through the vitreous humor, and then hits the retina. And the retina takes this information and passes it through the optic nerve to the brain. Here's a blown-up diagram of the retina. All right, so here's that same diagram I showed you, oriented in a different direction, as well as the actual histology of the retina. So light comes from this direction, passes through the retinal ganglion cells and the retinal interneurons, and strikes the rods and cones, which are the photosensitive cells in the retina. And those are these right here. And then the rods and cones take this information, pass it to the retinal interneurons, which process this information and send it to the retinal ganglion cells, which process this information some more, send this information down their axons, which converge at the optic nerve, and this information goes to the brain. All right, so you can categorize responses to light in 2 major ways. So first of all, there are image-forming responses to light, and this is how animals detect shapes, colors, and motion, and we call this vision. But there are also non-image-forming responses to light, and this includes, but is not limited to, synchronization of the circadian clock to the day-night cycle, light therapy for people suffering from winter depression, and also pupil constriction. All right, so while Professor Provincio was a graduate student, he was working with a strand of mice called RD mice. And what these graphs show are the histology of both wild-type mice and RD mice. Here you can see the ganglion cells, here are the interneurons, and here in the wild-type mice you see the rods and cones. And as you see in the RD mice, the rods and cones are completely absent. So as far as they knew, these mice were blind. And Professor Provincio and others eventually discovered some interesting things about these animals. So I'm going to show you something that Rob Lucas at the University of Manchester discovered about these mice. And what this graph shows is a dose-response curve of light on pupillary light reflex, or PLR. So as you know, in the dark, pupil is dilated, and when light is shown into it, the pupil constricts. All right, so here are our wild-type mice. So as you can see, as you increase the brightness, the pupils constrict more, and eventually it levels off because the pupil only constricts so much. And they discovered something really interesting about these RD mice. As it turns out, these mice, which lack rods and cones, still constrict their pupils in response to light. However, the response is diminished, and as you can see, for any given decrease in pupil constriction, or for any given pupil constriction, you need more brightness for the RD mice. All right. And Professor Provincio and colleagues eventually discovered that 1 to 2% of these retinal ganglion cells express a photosensitive pigment called melanopsin. And this melanopsin confers photosensitivity on these cells. And it turns out that these cells are partly responsible, we call these now intrinsically photosensitive retinal ganglion cells, or IPRGCs. And these are what supply some of the information for non-image forming functions in animals. So let me go over these 2 pathways. So you have image forming pathway, or vision. So light strikes rods and cones. This information is sent to the retinal ganglion cells and then to brain sites mediating visual responses to light. Now for the non-image forming pathway, the rods and cones receive light information. Then they send this information to the IPRGCs, which I've mentioned are themselves photosensitive. And this information is passed to brain sites mediating non-visual responses to light. All right, so in the RD mice, you have no rods and cones, so they have no vision, but because these IPRGCs are photosensitive, they still have some information passing to the brain mediating these non-visual responses to light. All right, so Luebbert, this major Danish pharmaceutical company, last year sent us this compound, and they basically just told us that we think it temporarily disables the function or output of melanopsin. So what I've been doing for these past 6 months is evaluating whether this compound is actually effective at doing that. Well, why would you want to do this? Well, first of all, it'd be really valuable for research because you could evaluate the same animal with and without functional melanopsin. And there are non-image forming function related disorders, including winter depression and some forms of anxiety. So the more we know about melanopsin, the more we might understand about these disorders. And these are my basic questions. Does the drug effectively disable the function or output of melanopsin? And how long do the effects of the drugs last? And the way I did this was by evaluating pupillary light reflex. So this is the same graph that I showed you before. And now I've added a double knockout mice. So OPN4 is the genetic name for melanopsin. So in these mice, the gene is knocked out for melanopsin and the rods and cones are knocked out. So they essentially have no melanopsin and no rods and cones. As you can see, they exhibit no PLR whatsoever. Okay, so in order to establish whether the compound works, I used RD mice. So since the RD mice already don't have rods and cones, if I inject them with a compound that disables melanopsin, they should essentially resemble those double knockout mice. So their PLR should be significantly diminished, if not completely absent. And then to evaluate how long it lasts, I just measured PLR at 30, 60, 90, and 120 minutes post-injection. So this was the basic experimental protocol. I handled the mice two consecutive days prior to PLR measurement. And the reason why I did this was because PLR is heavily modulated by the autonomic nervous system. So if a mouse is frightened, for instance, the flight or fight response will be triggered, which causes the pupil to dilate. So I handled them in order to try to reduce anxiety before I did the procedure. So after I did that, and immediately before the procedure, I got baseline PLR measurements. I then anesthetized the mice with isoflurane and injected both eyes with 2 microliters of either compound or vehicle. And then I evaluated the PLR 30, 60, 90, and 120 minutes post-injection. And then the way I evaluated PLR, I basically had a digital infrared video camera. And so what I did is I took a video recording of their pupils in the dark, and then I turned a light on, shined a light into their opposite eye, and measured their constricted pupil. Then afterwards, I looked at the videos on the computer and measured the diameter using a software-based pixel ruler. And that's how I got the size of the pupils dilated and constricted. Okay, so these are the results of the experiment. What this graph shows is the fold decrease in pupil constriction of both these groups compared to all the mice pre-injection. So 4.5, for instance, would indicate that their eyes constricted 4.5 times less than the mice pre-injection. And what's strange is you would assume that the assumption was that the vehicle injected mice would be unaffected, but there appears to be some effect of the vehicle, either the vehicle or the procedure on these mice. And the compound mice appear to have an even greater reduced pupil constriction. Unfortunately, there's a great degree of variability in my results, as indicated by these error bars, and the results were not statistically significant. So there's no statistical significance of the effect of the compound on these animals, and there is also no difference between the vehicle and compound groups at any time after injection. All right. So I'd like to continue this experiment using a larger sample size, and I think I can also reduce variation in my results a couple ways. First of all, the injections themselves, you risk damaging the eye, either the retina or the lens or some other component of the eye. So just being more careful of that would help. And also, a proper restraint system would help. I was basically just holding these mice. And if I had a proper restraint system, it would make it easier to measure their pupils, and it would let you use presumably the minimum amount of force necessary to keep them in place. Unfortunately, the restraint systems that I know of and that we have in this lab, in our lab, obstruct the eye, so I couldn't use them. But that would be useful in the future. So I'd like to thank the Provencio lab, which include these three wonderful people, as well as Mike Selix in the Miniker lab for helping me with this research. And I'd also like to thank the NIH for providing funding. Thanks. Any questions? Yes.
If you go back to the slides where you show the fold reduction, yes, thank you. You're measuring the diameter
Right.
of the pupil, right? So what you're measuring is pi r squared.
Right.
I mean the area.
Right.
So going from an area, how do you get fold decrease in pupil constriction?
Okay. So
What does twofold decrease mean? If you went from 1 unit of area to a half unit of area, threefold 1 unit of area to a third unit of area?
Right. So what I did, I took the pre-injection mice, looked at the area dilated, and then I looked at the area constricted, and I used a fraction. So
is this the standard way of presenting that data? Why don't you just present it in terms of area?
Oh, using just the raw data?
Yeah, I mean just the area.
Yeah, that's a good point. There are a couple of reasons.
This is sort of, you wonder what's hidden in this.
Yeah, that's a good point. There are a couple of reasons why I didn't use the raw area. I would have liked to. But first of all, well, the main thing really is that for individual animals, since I didn't have a proper restraint system, I could have held them at slightly different distances from the camera. So the assumption was that it would be more reliable if I looked at the change for each animal rather than the raw.
You could still normalize it to full dilation for that animal. Or am I missing something?
Normalize it full dilation for that animal.
That's 100%.
Right.
And then you just measure the area relative to 100% for that particular animal.
Right.
Well, it's not a big deal. I was just curious if this is a convention in the field or what.
Yeah, well, I mean, I sort of did that because I used the fraction remaining for each animal after constriction. So for that animal, it's showing, like, I would have a fraction, like 1/4 would mean 1/4 of the pupil remaining after light is shown into the pupil. Does that make sense?
Thanks.
Mm-hmm.
The intensity response curve of this system is very fast, like 3-4 units. What intensity?
Oh, what intensity did I use? Yeah, that's a good
How
question.
much do you use?
Basically, the idea is I wanted a sufficient intensity to trigger melanopsin, but I didn't want to use like a really high intensity because if the drug wasn't 100% effective, which assuming it wasn't.
Half point is
Yeah, sure. It was about right here. Yeah, and I didn't want to use too bright of light because if not all of the cells were, whatever, disabled, then if you use sufficient amount of light, it would have achieved full constriction regardless. So it was kind of a, balance. I chose the intensity. Yes?
How did you decide on the dose of the drug?
Oh, yeah, that's another good question. So I just used the dose that Luebbert
It was written on the
recommended.
box.
Exactly. So, well, no, they gave us like basically, like a recipe for creating a compound, and I followed that recipe. But I mean, I'd really like to eventually do a dose response curve because possibly, using more compound would help. But I just haven't had the time, and I also don't have as much compound as I'd like now. So if I get more compound, I'd like to do a dose curve for the compound.
Okay. Thanks, Sam.